Welcome back, everyone. So all of this contraction that we've been talking about, all of this muscle contraction and tone and all of these good things, all of it comes from, or the way we get it essentially is we have to have a source of energy, right? We have to have a source of ATP if we want to get our contraction. So we can have some amount of ATP that is just stored in our muscle. And we use this up relatively quickly within just a few seconds. Um, we use ATP for a number of different things in the muscle contraction sequence. So one, we use it for contraction itself, right? We have to form the cross bridges, and you have to have ATP to split it. We also said you have to have ATP for relaxing the muscle as well, and that serves to break the cross bridges when ATP comes in and replaces the ADP on the myosin head. And then we also said you have to actively pump calcium back into the sarcoplasmic reticulum, so you have to use ATP for that as well. So muscle contraction is a very energy expending and very energy expensive process. And so we have to logically ask ourselves, where are we getting all of this ATP, right? So first location that we can get it from is what's called our phosphate transfer system, all right? So in our muscles, we have a molecule called creatine, right? And creatine is a energy storage molecule, essentially, that can bind phosphate, all right? So it can bind a single phosphate molecule, right? When creatine is bound to phosphate, you call it creatine phosphate, right? So we have ATP in our muscle contraction. We said that you take ATP and you split it, you hydrolyze it, and you break it into ADP and phosphate. That lone phosphate can then travel away into the sarcoplasm of the muscle cell and bind to creatine. And when it does so, it creates this creatine phosphate here. And then you have the lone ATP from whenever we split up ADP, right? So you have ADP and you have phosphate. And then you can add these two together and you get this creatine phosphate here. So this is what we see in resting muscle, right? Phosphate is bound to creatine. And then in the event that we need that extra phosphate to add it to ADP to create ATP, we can do that. And so this is what's happening whenever we contract our muscle, right? So we take this creatine phosphate and we add the phosphate to the ADP to create ATP. And because we got rid of the phosphate, instead of it being creatine phosphate, we just have creatine, all right? So we can continually generate this amount of ATP via this system. It does not last extremely long. There's an enzyme that actually catalyzes this reaction called creatine kinase. But we'll need to rely on other sources of ATP if we plan to use our muscles for any extended period of time longer than, say, 5, 10, 15, 20 seconds, something like that, right? So the other way we can, or one other way we can get oxygen, we can get ATP, is via something called glycolysis. Glycolysis is a form of anaerobic respiration, meaning you do not have to have oxygen in order to produce ATP in this instance. You'll talk more about this in an AMP2 course whenever you look at something like nutrition and metabolism. Glycolysis can occur with oxygen, but it doesn't have to. So you don't have to have oxygen. So it's considered anaerobic. Glycolysis produces 4 ATP total, so gross, but you only net 2 because you actually had to invest 2 into the system, right? So you had to add 2 ATP molecules, and then you get 4 out, and so you end up with a net of 2, right? You also end up with something called pyruvate. Pyruvate is a 3-carbon molecule, and we started with glucose, which was a 6-carbon molecule. So you take one 6-carbon molecule and you split it into 2 smaller 3-carbon molecules, and these are called pyruvate that you see down here. And these pyruvate molecules then enter into something called the Krebs cycle, sometimes called the citric acid cycle, which is aerobic respiration that requires oxygen. And then you move into something called oxidative phosphorylation, and this is where you make large quantities of ATP. All right. So glycolysis, you make a little bit more, but again, if you're going to be using your muscles for any extended period of time, you're going to need a different source for your ATP. And this glycolysis, this occurs in the cytosol, I should have mentioned, which is the kind of liquid, the watery substance outside of the nucleus in our cells. All right. So depending on what you're doing what type of exercise that you're performing, you can have, or you can rely on different sources of ATP. So if you're doing something very quick, you just need quick, fast energy, immediate source, you can rely on your phosphate transfer system, right? So you have some ATP that's just available. You have the creatine phosphate that can donate the phosphate to the ADP to create some extra ATP. If you're doing something very short, 5-10 seconds, something like that, right? If you're doing something a bit longer, maybe a minute, couple of minutes, you may need to rely on a source of anaerobic respiration for your source of ATP. So you may be relying on glycolysis, but you could also rely somewhat on your phosphate transfer system to supply you with a little bit of ATP there as well. All right. And then if you're doing something extremely long, you're running a mile, you're running 400-meter dash, you're doing a marathon, a triathlon, something like that, you're going to heavily rely on your aerobic pathway. So your Krebs cycle that you'll talk about in future classes and oxidative phosphorylation to create lots and lots of ATP. Okay. So here is a summary. You can see these. So we've got our direct phosphorylation here. So we're using our phosphate transfer system with creatine phosphate. I mentioned earlier this creatine kinase. Anything that ends, or any enzyme that is a kinase enzyme, this is a phosphorylating enzyme. It produces or it adds a phosphate molecule onto something else, right? So in this case, you're adding phosphate onto ADP to create ATP, right? You then have your anaerobic pathway, which is glycolysis, and then you have your aerobic pathway, which is what we generally think of as cellular respiration using the mitochondria, and you produce large numbers of ATP here. All right, so what we'll finish with here is talking about smooth muscle. And so the start of this lecture, we mentioned that this is called skeletal and smooth muscle, and we allowed a whole 2 slides for smooth muscle. So it is not at all the focus of our lecture, but we will mention it briefly. So smooth muscle is named smooth because it is non-striated, right? So it still has the actin and myosin that you see in skeletal and cardiac muscle, but it is organized differently, all right? Our smooth muscle cells are spindle-shaped or tapered, whereas our skeletal muscle cells were more fibrous and cylindrical. So tapered and spindle, they're kind of thicker in the middle, and then they come down to a point on the ends. They're typically mononucleated. They're involuntary, so we are not able to consciously control the contraction of our smooth muscle cells. They do not have T-tubules, but instead they have something known as caveolae. So remember, T-tubules were these large extensions of the sarcolemma deep to the interior of the cell. Our smooth muscle cells do not have that, but they have something similar called caveolae, all right? Again, they're not organized, the myofilaments are not organized into sarcomeres, so it appears smooth. And then they also have something called dense bodies. And these dense bodies are analogous to the Z discs that we mentioned with our sarcomere in skeletal muscle. So if you recall, one sarcomere was one Z disc to another Z disc, and everything in between that was the sarcomere. So for all intents and purposes, you can think of smooth muscle cells, the dense bodies are like the Z disc. So from one dense body to another one, you have some myofilaments in the middle. However, there's another difference here. So in skeletal muscle, we had our Z disc, they're all in a line, right? They're all one after the next, parallel, right? In our smooth muscle cell, you have dense bodies that are kind of oriented all over the cell. So you have one here, you have one here, you have ones above it, you have ones below it, you have ones to the side. And so what this results in is when these myofilaments contract, these 2 dense bodies move closer together, but so do these 2, and so do these 2 over here in this one. So instead of getting a simple shortening and elongating of the muscle cell, of the smooth muscle cell, like we have in skeletal muscle, you get more of kind of a crumpling of the cell, right? So you have all of these dense bodies moving closer together, and so you get this nice contraction of the cell, but it's not really a simple shortening and elongating. It's more of a kind of shortening and crumpling. And then, of course, whenever they relax, they'll move away from each other, just like the Z disc did in our skeletal muscle, right? So we have so we have smooth muscle in a number of different areas in the body. So lining hollow organs like our GI tract for peristalsis. We have it responsible for some involuntary movement, so like in our gut, but also for things like our erector pili muscle that causes goosebumps. We have it in our respiratory passageways as well. So leading to bronchoconstriction and bronchodilation, so changing the size of our bronchioles leading into our lungs. You also have it in large organs like the uterus, gallbladder, and urinary bladder from movement of things in those respective organs as well, right? So this is where we will end. So this is the end of our Chapter 9 lecture. If you have any questions at all about any of this material, please do not hesitate to ask, and I will see you in the next video.